Welcome to Virology. I'm Vincent Rackaniello. I'm going to be your professor for this entire course. If you want to understand human biology, if you want to understand human health and disease, you need to know about viruses. And that's why I teach this course. I'm a professor at the Medical School of Columbia University. I've been there for over 35 years. I have a lab that researches viruses. And about 8 years ago, I realized there was no virology course taught down here on the Morningside campus, and that was a shame, because not only are viruses incredibly important for our health and well-being, as well as that of the entire planet, and I'm going to show you that in this course. They're incredibly interesting and fascinating entities. And I decided you needed to learn about them, so that's why I teach this course. I should tell you that as a medical school professor, I don't have to teach anything. All I have to do is do research. It's very different up there than down here. So I'm doing this on my own, voluntarily, because I love viruses, and I want you to love them too. I'm sure you'll love them, and I'm going to tell you everything I know about viruses over the next 25 lectures. We're going to go in great detail, and I think you're going to really like it. I'm going to try and give to you the passion that I have for viruses. We live and prosper in a world that is full of viruses. And if you learn one thing in this course, you're going to learn that you're full of them. Right now, you're sitting here. They're coursing through your bloodstream. They're in your intestines. They're in your lungs. They're everywhere in your body. They're on the surfaces of this room. They're floating in the air. You can't escape them. They infect every living thing on the planet. We've found viruses that infect just about everything, but not all, because we haven't looked hard enough. And we carry genomes as part of our genetic material, as you'll see. The whole game with viruses is numbers. They make huge numbers of progeny, and maybe a few of them end up infecting a cell and moving on. Because life is hard out there for a virus. The numbers of viruses on Earth is amazing. So just in the oceans alone, 10 to the 30th bacteriophages, that is viruses that infect bacteria. In the oceans of the world, about 1 million per teaspoon. Next time you go swimming in the ocean, you take a little water in your mouth and you spit it out at your friend, you are spitting at him or her viruses. They're harmless, they're bacteriophages, but they're there and they're infecting other organisms in the ocean. Now, 10 to the 30th is a huge number. You can't even begin to imagine it. Here on the left, by the way, is the structure of a bacteriophage solved by crystallographic and cryo-EM methods. And they have very different appearances, as you'll see. First of all, the weight. A bacteriophage like that weighs about a femtogram, 10 to the minus 15 grams. So if you multiply that times 10 to the 30th, the biomass of all these viruses in the oceans exceeds that of elephants by 1,000-fold. So that is something you can think about. Elephants are big. And as this course goes on from year to year, there are fewer and fewer of them, unfortunately, but they weigh a lot. But all the phages in the oceans weigh more. But even more interesting, if you put these phages head to tail, they go 100 million light years into space. 100 million light years. It's amazing. It's way outside our solar system. So that is amazing for something you can't even see. That's the point. There are so many of them on Earth, just in the oceans, that they line up and go a long way. Now, what's the source of all these viruses? Well, in the oceans there are many mammals that inhabit, like whales and dolphins and so forth, and they're infected with their own viruses, and whales in particular have small RNA viruses that infect them. They're called caliciviruses. We'll talk about human versions of these later on, and these caliciviruses, these whales excrete them at about 10 to the 13 particles per gram of feces a day. 10 to the 13 per gram of feces per day. It's a huge amount of virus for just one whale. And every mammal in the ocean is infected, every fish in the ocean, and all the way down to the unicellular organisms. They all have their viruses, and they are multiplying in the oceans. And these viruses can make the whales sick, by the way, and there's some evidence that they can jump into humans as well. You ever go to those shows where you sit in the front row and the killer whales jump out of the water and they splash and the people in the front row get wet. Well, they're getting showered with killer whale viruses. That may not be a good idea. So here's more information on the viruses in the ocean. There are more viruses in a liter of seawater than there are people on Earth. Next time you go, take a liter and look at it and think about that. It's amazing. What is this virus in the ocean? Here we have two pie charts illustrating it. One on the left is biomass. If you look at prokaryotes, protists, and viruses, you can see the prokaryotes are the biggest by biomass, okay? But if you look at abundance, particle number, the viruses win. 94% of all the particles in the oceans are viruses. They're just teeming with viruses, yet in many places that water looks very clear, of course, because they're so tiny. And these viruses are incredibly important. There are 10 to the 16th HIV genomes on the planet right now. We know how many people are infected. We know roughly, on average, how many genomes there is per person, and that's what it adds up to. And that's important because it's a huge genetic diversity. In this diversity is resistance to any antiviral we already have. We have over 30, and any that we would ever make in our lifetimes. That's how big that number is. So you're all infected. You probably sort of knew that, but I'm going to really emphasize it in this course. You are all infected with some kind of herpes virus, for sure, okay? We've probably got at least two of them, and there's herpes simplex type 1 and 2, varicella zoster, chickenpox virus, human cytomegalovirus, Epstein-Barr, and then human herpes viruses 6, 7, and 8. And these are really interesting. You get them quite young in age, most of them, from your parents. You know, your parents love you, they kiss you early on in your life, and they have herpes viruses in their saliva, and they're passing them on to you. And they stay with you forever. Once you are infected with any of these herpes viruses, you cannot get rid of them. And I'm very amused if you look at the herpes or the latency lecture for this course on YouTube, all these people writing how they were cured by a doctor so-and-so of their herpes. Impossible. Cannot be cured. We have no way of curing you of any herpes virus. You're infected for life. And I like to say, unlike love, herpes is forever. As you know, we have a microbiome. We have bacteria that colonize every organ system in your body, everywhere. It's really remarkable. This is an old slide to illustrate some of them in different places. We have a skin microbiome that varies from your left to your right hand. And we have it on our tongues, on our faces, and so forth. And this is, we're learning, is very good for us to have this microbiome here. We also have a virome, but the virome doesn't get a lot of attention. I don't know why. I think it's just as important as the microbiome. Maybe because it's harder to enumerate the virome. To study a microbiome, all you need to do is extract some nucleic acid from a sample. You could take a skin swab if you want to study the skin microbiome, and you extract nucleic acid and sequence the ribosomal RNA, and you have to do a very small amount of sequencing to get an idea of what bacteria are present. It's pretty easy because ribosomal RNA is in common among bacteria, but viruses don't have a common sequence that we can use to identify viruses in a sample, so we have to sequence the entire genome. And so in these experiments shown on the board, on the right you have an experiment where they took human blood, and they extracted nucleic acid from it and did very high throughput sequencing, a technology we call deep sequencing or high throughput sequencing. It used to be called next generation, but it's no longer next generation because it's here now. So I'll try not to use that phrase. But you can know a great deal and very, very high complexity about what nucleic acids are present. And look at this. 67% of the sequences in your blood are viral. Okay, all of you, if I did this experiment on you, you'd probably get something similar. 9% bacteria. Human, 3.6%. The minority, and that's amazing, right? And then unknown is 15%. There's always an unknown in all of these sequence analyses where you take a sample and just do bulk sequencing and try and see what's there. The way you identify what's in a sequence, of course, is by comparing it to a database. There's a big database out there of all the sequences that have been collected, and you use computational methods to compare what you get against it. And often you get no match, and that's what we label as the unknown DNA. On the left is a more detailed study of the human virome in every part of the body that's looked at. And you can see it's divided here into blood, digestive tract, nervous system, various teguments, respiratory and GU tract. There's a unique set of viruses, and the different viruses that are listed here, DNA and RNA viruses, that are found at each of these locations. So these are all healthy humans. They're not sick. We have a huge virome in us, and it's always changing. And what we're trying to do now is to understand if it's beneficial or not, and if so, how we can manipulate it. Bacteria you can get rid of by treating people with antimicrobials, but you can't do that with viruses. We don't have many antivirals that will simply wipe out your virome, and then we can see what happens. Now, I mentioned earlier that we have viral genetic sequences as part of our DNA. And this is really remarkable. It's shown here on this pie chart of the human genome. Your genome, 3.2 billion bases, and it's divided up into the different parts that it encodes here. The protein coding genes are 1.5%. So the minority of your DNA actually encodes proteins, 20 to 30,000 different proteins or so that make you up and allow you to function. But the rest of the genome is all sorts of stuff that we're just beginning to understand, like introns, heterochromatin, duplicated sequences, interspersed elements like lines and signs, which we'll talk about later on. But here, for our purpose today, this 8.3% LTR retrotransposons. Many of these are integrated retroviral DNAs. So a virus called retroviruses that is an RNA virus, converts its genome into DNA, and then integrates into the infected cell. And so most animals on Earth have experienced retroviral infections, and those DNAs integrate into the chromosomes of the animals and are passed on to their children and so forth. So we have a good fraction that is with us permanently. We inherited it from our parents. Probably they were there, many of them were there before Homo sapiens even emerged on Earth. We'll talk a little bit about that later on, and we'll talk about some of the amazing aspects of this, which is what are they doing here? What are these sequences doing here? Most of them don't make infectious viruses, but it turns out that many of the proteins have been exapted. That's a word which means you take a virus protein that you got and you use it for your own purposes. Exaptation. And so we have actually exapted a number of these retroviral genes for our own use over the years. So we really are viral. And it would be an interesting study to take out all these endogenous retroviruses and see what would happen. People have done this in pigs, because as you may know, people want to use pig organs for transplantation. We don't have enough human people dying to supply all the organs we need for transplantation. I know that's a morbid statement, but that's the reality. So we're turning to pigs, for heart valves and so forth. But the problem with pigs is that they have lots of endogenous retroviruses, and they actually make infectious viruses. So a group at Harvard has removed all the endogenous retroviruses from pigs. And so far they're okay, these pigs, but not 100% of the offspring are well. So it may be that you actually need some of these sequences. We'll talk about those more later. So this should give you a sense that we are accosted by viruses on a daily basis, just as you are now. But most of the viruses that infect us, they don't really infect us. They don't really have an impact on our health or well-being. They may have a positive impact, which is something that remains to be shown, but they don't seem to hurt us. And why not? Why can we live in a sea of viruses, a cloud of viruses, and be okay? Well, there are a couple of reasons I'd like to set forth. There are probably more. And the first one is that most viruses just pass through us. So here's one for the coleslaw lovers. If you like coleslaw, coleslaw is made from cabbage. And a recent study was done which showed that in the Washington, D.C. area, cabbage purchased from 5 different supermarkets were contaminated with baculoviruses. This is a baculovirus. It's a rather large virus that infects a variety of insects. And in each serving of coleslaw would have up to 10 to the 8th particles of baculoviruses. So when you're eating coleslaw, you are definitely eating baculoviruses. These viruses got there because of insects that were infected with them crawling over the cabbage before it was harvested. And one of them is this cabbage looper, it's called. They're infected with baculoviruses. They rupture, they die on the leaves, and the baculoviruses contaminate the leaves, and you can't get them off. You could wash them forever. You can't get them off, but it doesn't matter because you eat these and they pass through you and they do not infect you. They are insect viruses. And people have done metagenomic analyses, take total RNA or DNA from any place, your body or any place in the world, sequence it, and 91% of the RNA viruses in human feces in one study were plant viruses. And so here is an example of an insect that feeds on a plant that gets into you, but also many different plants that you eat are full of viruses. And one of the most common plant viruses infects a pepper mild mottle virus. And that's the most abundant human fecal virus. So we love our pepper. The next time you eat pepper of any kind, just think it's probably full of viruses because the plant that it was made from is full of viruses. But again, these pass through you. They do nothing to harm you whatsoever. Maybe they help in some way. We don't know. People have not done the right experiment. So one idea is that viruses pass through you. The other is that there are beneficial viruses in every species. This is a hard thing to prove in people because you can't really do many experiments with us. You can't take away viruses. You can't add them back very readily. You can do things with animals or plants. So here's an example of a beneficial, or what I call a good virus, of plants. And this is a grass called Dicanthelium lanuginosum, which is famous because it grows in very high temperatures. If you ever go to the hot springs around Yellowstone or any of those parks with hot waters, the grasses that grow near the hot water are pretty temperature resistant. They can grow at high temps. Here, 55 degrees. You can bring these in the lab and show that they'll grow at 55 C. It's pretty hot. Now, the ability of this grass to grow at high temperatures depends on a fungus that infects it. And here the fungus is Curvularia protuberata. It is a fungus that is inside the plant, and that fungus, if you take it out of the plant and then try to grow the plant at 55, the plant will no longer grow. That's shown here by this non-colonized, withered up plant. However, what really makes the difference is that there is a virus inside of the fungus. So there's a fungal virus that confers this thermotolerance. If you take the virus out of the fungus and put just the fungus in the plant, it is not able to grow at high temperatures. So this is a good virus. It's not killing the fungus, it's not killing the plant, but it's benefiting the plant and allowing it to grow at high temperatures. And this is just a form of some kind of selection and evolution that has occurred over the years. It's a mutualism involving multiple organisms, and we're going to see lots and lots of examples of that. So it's reasonable to extend this to people and say there must be viruses in us that are beneficial. We do have some evidence in mice, which we can manipulate experimentally. And here's what I think is the best evidence in a mammal for a beneficial virus. And this is an enteric virus called norovirus. It's a murine virus. Related noroviruses can infect humans. We'll talk about those later. But this is an RNA-containing virus related to those viruses I told you about that infect whales. In this experiment, they found that first they removed all the bacteria from the mouse by treating them with antibiotics. And then you have germ-free animals and you have conventional animals that have a full complement of bacteria in their intestines. So these are sections of the villi of the small intestine. And you can see this is a normal villus. And on the germ-free animals, the villus is malformed. So you need bacteria in the gut to allow the villus of the intestine to form properly. But what also happens if you take away bacteria is that the T cells in the gut don't mature properly. So this lower panel, the two lower panels are stained for T cells, T lymphocytes in brown. And in the conventional animals, there are good numbers of them, but in the germ-free animals, the numbers are very low. So both the immune system and the intestine itself, the development of those two organs requires the presence of bacteria. What was amazing in this study, though, was when they infected these mice with a murine norovirus, it partially restored the defect in the morphology of the intestine that had been caused by removing bacteria, and it restored the presence of lymphocytes. You can see now there are lymphocytes present. This is work done at NYU here in New York by Ken Cadwell's lab. I think it's the best evidence that viruses can be beneficial for mammals. And of course, we need to do similar experiments in humans, but we can't really do it. Someday when we have broad spectrum antivirals, we're going to have those one day, we'll be able to figure out if viruses are important for us. I think they must be. We have so many of them in us. We've evolved with them for so many years. They must have a function. So I think lots of the viruses in us are good. But really, in the end, the reason why all of you are pretty healthy, maybe you have some little influenza or upper tract viral infection, but you're pretty healthy. The reason is because you have a great immune system, which is diagrammed here. And this really takes care of any nasty viral infection that gets out of hand, or it should take care of it. If your immune system is in great shape, you will get rid of virus infection. However, if your immune system is down, if you are immunosuppressed for whatever reason, you have an organ transplant, you get immunosuppressive drugs so you don't reject the organ. If you get measles, measles will immunosuppress you. It replicates in immune cells and causes you to be more susceptible to infections. And of course, HIV is the big immunosuppressor, replicating in CD4 T cells. If you are immunosuppressed in those ways and more, then the smallest viral infection causes a problem for you. So an infection, which is not really an infection in most of us because it doesn't cause disease, will be a problem if your immune system is down. So the immune system is really important for that. Now, in this course, we're not going to spend a lot of time talking about the immune system. Rather, we're going to talk about the interaction of your immune system with viruses. So some of you have taken Moshewitz's immunology course, I know, so you'll have a great immunology background. And I'll tell you what you need to know to understand the interaction of viruses with that immune system. But the best way to take this course is, of course, to take immunology first. Now, not all human viruses make you sick. I told you about plant and insect and other viruses that pass through you and don't infect you. But we do have human viruses in us, and they're not making us sick. They've evolved with us for many years, and so they have evolved to be non-pathogenic. And we will talk about some of the mechanisms involved in this course. And here's an interesting virus that is present in pretty much all humans. It's a small double-stranded DNA-containing polyomavirus. Here they are shown here. Most of the population of human population of the planet is infected with these viruses. Again, if you have a good immune system, no problem. If you're immunosuppressed, these can cause serious infections of different organs. And we'll talk about that. But these viruses are spread within families. So the family you grow up in, you get the particular polyomavirus that's in that family, and a different family in a different country gets a different one. So you can actually look at who has what kind of polyomavirus and track human migration over the years. And that's what this map shows you. The dotted line is the migration of humans out of Africa into Europe and Asia and the Americas. And the solid line is the migration of humans based on infections with polyomavirus. As you can see, it's way more detailed. We can really track a lot more than the human genome sequence. So these are viruses that normally don't make you sick. And why is a good question, which we'll try to explore. So those are just some tidbits of what we're going to be talking about in this course. I think viruses are amazing. And more importantly, maybe for you, for those of you who are science majors or who want to have a career in some kind of medicine, virology is an integrative science. It's not like chemistry, where you study chemical reactions and that's it. It's not like other sciences, where you study that and the relationship to other sciences isn't clear. With virology, you need to know pretty much every science to understand viruses. You need to know molecular biology, cell biology, epidemiology if you want to study disease, ecology for sure, if you want to see what viruses do in the environment. And so I think by studying virology, you learn about all these other things. And many students who have taken this course in the past come up to me at the end and say, now I understand biology. It all makes sense. It didn't make any sense before. Now I understand cancer. So I think this actually should be required for everybody to learn some kind of virology, because I think it makes all other science interesting. Of course, that's not going to happen, but at least you're here and you can learn this. And really, I think it will clarify lots of other aspects of other sciences that you may not have thought about. So what I want to do in this course, before we move on to talking about what is a virus, I want you to see the big picture. So this course is not arranged by virus. Lots of professors teach virology courses by virus, and that's the easy way out. You don't learn anything when you take a virology course where lecture 1 is herpes virus, lecture 2 is polio, lecture 3 is influenza. All you're going to learn is about a collection of viruses, and you're not going to integrate it. What I want to teach you is the big picture of how all viruses work. That's hard to do. And I can only do it because I've been thinking about these things for 35 years, frankly. I don't think it's easy for anyone who has not worked on viruses for a long time to teach this kind of course. So, big picture of virology, and I want you to think of it as an integrative discipline, not a collection of facts, but putting them all together. And as we go through the lectures, which are based on principles, not viruses. There's only one virus that gets its own lecture in this course, and that's HIV because it's really important. But no other virus gets its own lecture because I want to teach you about the principles of virology, of diseases, and so forth. So you're going to learn the fundamentals, which frankly frighten most people. Now, many of you may come in here being scared of viruses, and you hear about an outbreak of Ebola or MERS coronavirus or even Zika, and you get scared. By the end of this course, you're not going to be scared anymore. You're going to understand what's going on. Because it turns out, most of your information on viruses you have gotten from the press, from, the web, news, if you watch any news on TV, etc., and they're not right on most things. This course is going to teach you pretty much the right things about viruses, I hope anyway, and you're going to end up being really informed, and you can tell other people the right things as well. I get lots of emails from former students who say, now I understand this outbreak, I understand what's going on, and I see how the press is exaggerating it. Talking about the press exaggerating, I have a number of pet peeves that I like to complain about, and you'll have to bear with me when I do that. You can tell when I get on my soapbox. And one of them is the coverage of virology. It's not just virology, it's other sciences, but I'll pick on virology because that's my field. The coverage by the general press. They often don't get it right, and they can't because they're on a quick schedule to get things out rapidly. They don't always check their sources, and they don't really understand. And of course, more importantly, they haven't had a course like this. So if any of you are ever journalists one day, you're going to have learned a lot that'll help you. So here is a CNN cap I took many years ago, 2009. We had an outbreak of a brand new influenza virus, H1N1 swine flu. And here, this fellow is reading off these bullet points. These are findings from an animal study done with this strain of influenza virus. So ferrets are a good model for studying influenza virus. And so this study was in the New England Journal of Medicine. So they decided to cover it here, but they didn't say that this was an animal study. They said this is what the flu is going to do. It's going to ravage your lungs. It's going to spread through your respiratory system and cause lesions. It turned out that the ferret studies way over-exaggerated what happened in people. In people, this turned out to be a pretty mild infection. But they didn't give any of the caveats here, and they scared a lot of people. Of course, if they scared them into getting a flu vaccine, that would have been good. But a lot of people don't understand this sort of information, and I want to teach you to be able to deal with this and all the other misinformations that come out there. Which statement is true? All viruses make us sick and can be lethal. B, our immune system can manage most viral infections. C, humans are usually infected with one virus at a time. D, the press is usually correct in their virology reporting. And E, our immune system cannot handle most viral infections. 99% of you got B, our immune system can manage most viral infections. That's correct. All viruses make us sick. No, and you got it. You nailed it. Very good. One virus at a time is wrong. Press is usually correct. That must be a journalism student who said that. Their immune system cannot handle most viral infections. So what is a virus? This is my definition of what is a virus. An infectious, obligate intracellular parasite with genetic material that's DNA or RNA. It's surrounded by a protein coat, and sometimes it has a membrane around it as well. And this is a definition that I've modified over the years as we've learned more. It's the simplest that I can get it so far. So infectious is obvious. It's something that can go from host to host. And by infection, we mean entering the cells of that host. Obligate intracellular parasite, that's really important. That means that the virus has to get inside of a cell in order to reproduce. If I have a tube full of pure viruses here, it will do nothing unless these viruses encounter a host cell. It has to get inside of a cell in order to multiply. So that's obligate intracellular parasite. And of course, parasite part means that the virus is causing some harm to the host. Now, that's an old definition, and there may be cases where a virus is infecting a cell, but you know what? It's always causing some damage because viruses completely depend on the metabolism and the molecular biology of the host in order to multiply. So even in viruses that don't cause any disease in you, there has to be some aspect of parasitism. And then we have DNA or RNA, which is really unusual. Viruses are the only RNA-based life forms that we know of on Earth. We'll talk about why that might be later on. And then they have, in the simplest form, they have a protein coat. So some of these virus particles here are just made of a protein shell with nucleic acid inside. And some of them also have a membrane around them, like this virus here. We'll talk about the structure and the principles involved in that. I just want to tell you that there are some virus-like elements which we call viroids. And they just infect plants. And they're just naked RNA. There's no protein on them. It's naked RNA. And what's even more amazing is they don't encode for anything. They don't encode for any protein. And we're going to talk about those. And they may be ancient versions of viruses. Now, because they are obligate intracellular parasites, or another way I like to put that is obligate molecular parasites. They depend on the machinery of the host cell, translation, metabolism, energy production, and so forth. And we'll talk about this in some detail. Whatever you learn about a virus reveals something about the host as well as the virus. And so molecular biology in its old era, in the 50s and 60s and 70s, people used viruses to understand cells because cells were too complicated and viruses offered pure genomes and relatively simple genomes that could be studied. And a lot of the major fundamental aspects of a cell that you have learned in biology were discovered through virus study. For example, splicing of mRNA, that was studied in a virus-infected cell. And many, many other aspects of the cell learned from viruses. Nowadays, we have really powerful experimental tools, and we can study cells on their own right. But viruses, for many years, they still continue to be, reveal something about the host. Now, is a virus living? This is an interesting question. Some people say, I don't care, but I say it's interesting because it makes you think about not only what constitutes life, but what is a virus all about? So I have a poll on my website, which has been there for a number of years. You can see I've got so far 6,300 people who have answered the question. Is it yes, no, in between? I don't know. So you can see people are pretty much split, yes, no, in between. But I think they're a little bit of both. I think and I've thought about this for many years, and I think this is the perfect solution. A virus is an organism with two phases. So a virus can exist as an infectious particle, or what we call a virion. I'll use this name a lot, virion, and I use it to mean the infectious virus particle. So there's one phase of infectious virus particle. So here's a polyomavirus, just to illustrate that. It's a protein shell with DNA in it. How could this possibly be living? It cannot do a thing on its own. So this is non-living. That's the non-living phase of the virus. The virus is an organism. It's the virion, and then the infected cell. This virus particle infects the cell. The infected cell is living, and the virus completely takes over the cell to make more virus particles. All right, so this is clearly a living entity, this virus-infected cell. So that portion of the virus is living. The infected cell is alive. The virion is not. I don't see how you can say that the particle is living. But in fact, when most people say viruses are living, they're imagining the infected cell part of the replicative cycle. And so this deals with that. So it really are two phases, one of them living and one of them is not. Now, another warning for you, and I will give you some warnings throughout this course. Please avoid anthropomorphic characterizations of viruses. Viruses are inanimate objects. They are completely passive. They have no intentions. They have no feelings. And they sense nothing. They're completely passive objects. They do not think. They do not employ, insure, exhibit. But this is, in fact, how many people describe them. But in reality, they don't achieve their goals in a human-centered manner. So and the reason this is important is because if you start to make assumptions about why a virus is doing something, you're applying your human bias to the perspective. The virus must be evolving in this manner because it wants to evade the immune system. When in fact, you don't know what the reason for any viral behavior is. So you're putting a human bent on what viruses do, and that's wrong because viruses are not human. They're passive objects with no memory or emotion or anything. They are just bags of nucleic acid that infect the cell, and then the cell provides an environment for reproduction. So I will try and avoid this. In the first few editions of our textbook, we had lots of examples of that, and we think we purged them all. But viruses are passive agents. They don't do things on purpose. They are at the mercy of whatever environment they're in. Now, part of the definition of viruses many years ago was that they were very small. In fact, that definition I showed you had very small included in it. They're pretty small, but I wouldn't say they're very small at all because we've discovered some really huge ones. So this is a picture that tries to give you the scale of virus particles. Here's an E. coli at 100,000 x, and there is a bacteriophage particle infecting E. coli. So you can see the size of the phage with respect to E. coli. C is tobacco mosaic virus. It's a rod-shaped virus, the first virus discovered. Above it is HIV-1 and D. And then there's a panel with other entities here, which is expanded a millionfold to the right. And there we have some actin and myosin molecules and a variety of enzymes. And here in H is a virus particle, a rather small one, a rhinovirus or a poliovirus. Above it is a ribosome. In G, in E is a tRNA molecule, and F is an antibody, and then A is a carbon molecule. So you can see the smallest viruses approach molecular size. And here's another slide to just give you an idea of the range of sizes. Here at the bottom left is a cell. And here are two different kinds of virus particles outside of the cell. The bigger ones are herpes viruses. They're about 200 nanometers in diameter. And then next to them are polioviruses, which are about 10 times smaller. And so viruses occupy a narrow range of sizes, and you can't see them in a microscope at all. You need an electron microscope or more powerful ways to be able to visualize them. If you want to know how many viruses fit on the head of a pin, there's actually an animation online that this comes from that you can zoom in or out on. And here's the pin with a strand of hair lying on top of it. And it's about 2 millimeters in diameter, or 2,000 microns. And we zoom in on this little square here. That on the left is a dust mite, which can hide under your beds and so forth and give you allergies. But to the right of it, which you can barely see on the left panel, here it's zoomed in. Here's some red blood cells. There's a lymphocyte and a yeast cell around here as well. And then here are some bacteria in green and yellow. And then the viruses are in the middle. The biggest one is Ebola virus, which you can see, but there are also rhinoviruses in there. You can't see them in this view. So the answer to how many rhinoviruses would fit on this pinhead is 500 million. And that's about what you would sneeze out of. If you were infected with a rhinovirus, which gives you an upper respiratory tract infection, you would be sneezing out hundreds of millions of rhinoviruses. And that's why they're so effective at infecting other people. Now, as I said, we used to think viruses were really small, but a number of years ago, over 10 years ago now, scientists discovered a huge set of viruses. We now call them giant viruses. And here was the first example, made the cover of American Scientist in 2011. These are mimiviruses. Biggest virus, at the time, the biggest viruses known. You can see comparison rhinoviruses, which we just talked about on the head of a pin. Here's HIV-1, a little bigger, but Mimi dwarfs them all. And these are pretty big viruses. Here they are in an infected cell. And some of these are so huge you can actually see them in a light microscope. And in fact, Pandoravirus was discovered in 2014. It's 1.5 microns long. And this is a light micrograph of mimivirus particles. So just a light microscope in the laboratory, which we usually use to look at tissue culture cells. You can't see any virus in it, but these Pandoraviruses, which have a very unusual morphology. As you can see, they're filled with double-stranded DNA. They're huge, and the genomes are huge, over 2 million bases in length, way, way more than the biggest ever known up to that time. So these challenge what we think is a virus, and we'll talk about those later on because they're really interesting. Now, an important fact that's going to stay with you throughout this course is how viruses reproduce when they infect a cell. They don't do binary fission. You may be familiar with how that works with our cells that divide, or with bacteria or yeast cells. You start with a single bacterium. It divides to make 2 and 4 and 8, etc. That's binary fission. So when you put a bacterium in a broth, it begins to divide. You have a logarithmic phase of growth, and then when the nutrients in the medium are expired, the bacteria stop growing. Viruses don't grow that way. They do not split, and that should be intuitive that a virus particle couldn't split. It has no way of doing that. But rather what we do with viruses, they make preformed components. So when a virus infects a cell, gene expression occurs, mRNAs are translated to make the proteins that are needed to assemble new virus particles. Okay, you make the parts and assemble the final product. And that's why the growth curve of a virus is very different from that of a bacterium. Bacteria, again, you put a single bacterium in a broth, it starts to divide immediately. When you infect cells with a virus, so on top is a growth curve of a virus, we're looking, with time, we are looking at the number of infectious particles produced. There is a period when nothing happens. It's called the eclipse period. There's no virus detectable. Very much unlike with bacteria, where you can see bacterial division right away. There's this eclipse period, and then at some point after you've infected the cells, and that point varies according to the virus, you then have the production of infectious virus. And then the cells are killed and no more virus is produced. That's called the burst, or the yield. But this eclipse period is very important because during that time, all the parts are made to make new infectious viruses. They're not yet assembled, so there's no infectivity detected, but they're being built. So very, very different from the way that bacteria work. All right, how old are viruses? The latest data, which came out last year, indicates that you can find some retroviruses, these viruses that integrate their genome into that of the host cell. So you can sequence the genomes of living organisms and see how these integrated retroviruses are evolved. And you can backdate by molecular clock techniques. We'll talk a little bit about that later, and estimate how old these integrations are. So the oldest one so far is about 450 million years ago, suggesting that there is a marine origin of these retroviruses. So that's Ordovician period right here. So life began, 600 million years ago, 450, the first fish and plants. We already had the Cambrian explosion, and the retroviruses seem to have, at least at a minimum, originated at that point and infected before jawed vertebrates arose in the ocean, infected individuals like this one here, these cephalopods. But I think that they actually originated before life on Earth. I think if you think of a virus as a replicating nucleic acid, I think many people agree that self-replicating nucleic acid-like molecules arose somewhere on Earth in the early days before cellular life. And I think those were virus-like entities. Even though there were no cells, you can imagine there was enough material for these molecules to self-replicate. And then from then, cells evolved. And so many people think that life evolved from viruses. So we truly are viral, I suppose, if that's correct. Now, moving more recently, we can find references to viral diseases in human written history, 700 BC. Rabid Hector is a reference in the Iliad, possibly referring to rabies virus. And of course, we didn't know anything about viruses back then, but the disease it makes someone rabid. And here, 1580 BC, this Egyptian carving shows a priest with a withered leg, which is interpreted to be caused by poliovirus. So many human viruses were around a long time ago. We know smallpox, for example, was around a long time ago as well. We can see in the written history records of infections that resemble infections we know today. Immunization against infection was practiced long before we understood what the agent of these diseases were. For example, variolation was practiced in the 11th century in China. At that time, we believed that smallpox was extensively found throughout the country, and it was found just empirically that if you got infected with this disease, they didn't know what it was, you had a chance of dying, but those who survived were never sick again. So very smart people made this observation. So then they started the practice of variolation, which is to take smallpox, as you know, causes pustules on the skin. Take one of those pustules, grind it up, and here you can see it's being blown into the nose of someone, or sometimes they scraped it into the skin. So basically they were infecting them, unknowingly they were infecting them with the virus. This carries about a 30% lethality, so it's not a great vaccine, but that's what they had, and those 70% of the people used it. And this practice came to Europe via Lady Montagu. She was the wife of the Turkish ambassador to Britain, and she saw this being practiced there and brought it back to the UK. And eventually it came to the US, the colonies, as well. There was extensive smallpox in the American colonies, and variolation was practiced as a way to get around it. But in the 1790s, Edward Jenner in England did the first experiments using proper vaccination, and that's really the first vaccine. Although at that time, he did not know what a virus was still. And I'm going to tell you when that happened in a moment, but the first vaccines were developed before knowing what a virus was. We'll talk more about Jenner later. To understand how we discovered virus, we have to think about microorganisms. Remember, for much of human history, we had no clue that there were anything smaller than what we could see. Another person, a tree, an insect. We thought that was all there was. And it took Leeuwenhoek, who made the first microscope, and was very curious. He looked at all his fluids, found giardia in his stool, and all sorts of things everywhere else. He saw the first microorganisms. And he established this idea that there are things we can't see that are in all sorts of fluid. And this got people really excited and thinking about it. And eventually, Louis Pasteur, of course, showed that there are bacteria that grow. They're microscopic. They're good for making wine. They are good for making cheese and fermenting and so forth. And they can also cause disease. And of course, Robert Koch developed the germ theory of disease. He showed that certain bacteria were responsible for causing disease and develops what we call Koch's postulates, the way to prove that a given organism could cause disease. So this brings us, to the late 1800s, but still no viruses on the scene. These individuals are all thinking about bacteria pretty much, bacteria and yeast. And that changed with this disease. This is tobacco mosaic disease. Tobacco was already an important agricultural product at the end of the 1800s. People were smoking it. And this disease causes a mottled syndrome on the leaf you can see here. And this is no good, apparently, for making smoking products. So lots of people throughout Europe were trying to figure out what it was, what caused it, and they figured it was a bacteria that caused it. So what they were doing is grinding up the leaves and taking that extract, and putting it on new leaves and transmitting the infection. And what they found was when you filter, so you make an extract of a leaf and then you filter it in a filter with a very small pore size. So in the old days, they were made of ceramic and they had very tiny pores. And if you filtered material through it, the bacteria would be hung up on the pore. And this is how people used to concentrate bacteria. And what they found with tobacco mosaic disease is that what was on the filter didn't cause disease in a new plant, but what went through the filter. If you took the fluid that went through the filter and applied that to a new leaf, it would cause tobacco mosaic disease. So it was something very tiny. And two people independently discovered this, Ivanovsky and Bayerink, and it was eventually called a virus because it went through the filter. They considered it to be a liquid. They didn't actually know it was a particle until many years later. They thought it was a poisonous liquid, and that's what virus means. Today we have very sophisticated filters that we use for the same reason, to sterilize a sample, remove the bacteria, and have virus in the fluid that flows through. So that was a plant virus. 1898 was the first animal virus. Foot and mouth disease. is to this day a serious disease of hoofed animals like cows. It causes lesions on the mouth and the feet, and these prevent them from being sold for meat. A very serious disease. It was shown to be a filterable agent, that is a virus. And the key concepts here, small agents that pass through the filter, but they will not replicate if you just keep them in a broth. They have to have a host on which to replicate. The filtrate that goes through the filter never increases in virus titer unless you put it on a new host, a plant or an animal in this case. And originally the filters were 0.2 micron filters. That defined a virus for many years. Of course, viruses are way bigger than that, so we have to throw away this definition that a virus is something that goes through a 0.2 micron filter because there's so many viruses that are bigger than that. Now, after that virus discovery proceeded, all of this was driven by disease. Yellow fever virus, we're trying to dig the Panama Canal. All the workers are getting sick with yellow fever. It's transmitted by a mosquito. It turns out to be a virus infection. So they get rid of the mosquitoes so they can complete the canal, but that was the first human virus discovered. Rabies, 1903, smallpox virus, chicken leukemia, the first cancer virus, and we'll talk about that in a lecture later on. Poliovirus in 1908. Another cancer virus in 1911 by Peyton Rouse, discovered here in New York at Rockefeller. 1915 bacteriophages, influenza virus not till 1933. This is pretty glacial pace of discovery. You know, every few years a new virus. Compared with today, the contrast is amazing. So let's first look at another question here. Which of the following is true concerning bacterial versus viral replication? A, viruses must assemble using preformed components. B, bacteria do not replicate via binary fission as viruses do. C, bacteria must assemble using preformed components. D, viruses do not have an eclipse period. E, viruses replicate by binary fission. 93% of you got A. Viruses must assemble using preformed components. That's correct. All the others are not right. Bacteria do replicate via binary fission, and so C is also wrong. Viruses have an eclipse period, and they don't replicate by binary fission. In the 1930s, the electron microscope was invented in Germany. And for the first time, people could see what viruses looked like. You could see that they were particles. There's tobacco mosaic virus. It was a rod-shaped particle. Here's a bacteriophage, rabies virus. And this dispelled the idea that they were liquids. They were clearly particulate. And nowadays, we know an incredible amount of detail about viruses. We can solve their three-dimensional structures, sometimes within weeks at high resolution, so we can make lovely pictures at high resolution where we can trace every atom in the protein chain of the particle, or at lower resolution where you can get an idea of the surface. Here we can know the chemical formula for poliovirus because we know the composition of the capsid and the sequence of the viral genome. We can sequence viral genomes at will. We have incredible amounts of information and amazingly powerful experimental tools to study them. Just a word on how we classify viruses, because I'm going to be throwing around some names in this course, and while you don't need to remember them, you might want to know what they are when I'm talking to you about them. So we classify all these different viruses by 4 different properties. The nature and sequence of the nucleic acid is the primary method now of classifying a virus. In fact, when you go out, if you take some water sample and sequence it and find some viral sequences in it, you can immediately classify those viruses based on the nucleic acid. You don't need to know anything else. But in the past, we've looked at the symmetry of the protein shell, whether there is an envelope, a lipid membrane in the virus particle, and the dimensions of the virus particle and the capsid. And the way virus classification is depicted is shown here. It's the typical hierarchical system. As you know, there are kingdom, phyla, classes. With viruses, we start with orders, which are collections of families of viruses, which have genera and species within them. So as an example, if we look at Ebola virus, the family is the Filoviridae family, which you can also say the filovirus family. There is a genus called Ebola virus, and then within it there are various species, including Zaire Ebola virus, Sudan Ebola virus, and so forth. The concept of a species with a virus is a little nebulous because species is originally defined by two organisms which cannot give fertile offspring when they mate, but viruses don't mate. So it's kind of weird that we call them species, but that has stuck. Today, you can take any sample you want, whether it's from a human, a mouse in the woods, an insect, the ocean, drinking water here in New York City. You can extract nucleic acids and find out what kind of virus particles are in it. And this is called metagenomics. And here's a great study published last year where came out of China. They extracted RNA from 220 invertebrate species. And you can see insects, crustacea, nematodes, worms, mollusks, etc. They just collected them all over the country and extracted RNA and sequenced it. And they discovered 1,445 new viruses. And this graph shows you the frequency of virus discovery in these different species. You can see some of them had more viruses than other in terms of the number of reads of sequence that you get. So anywhere you look, you can find viruses. And we haven't exhausted all the viruses that are out there yet. The more we look, the more we find. We find viruses that we can put into families, but we also find viruses that we've never seen before. And those sequences go in the database, and then the next time someone discovers it, it matches up, and you can say now it's a known virus. But discovery now is just driven by curiosity. We used to say, oh, here's a disease, yellow fever. Let's find out what causes it. Here's influenza. Here's a weird tick-borne disease in the Midwest that gives farmers fevers. And we still do that to a certain extent. That's disease-driven virus discovery. But most people now want to just be curious and know what's out there, know where in the world all the viruses are, what they do. And we have the technology to do that, which is really remarkable. Now, you may say, why do I care about how many viruses are out there or what kinds they are if they don't infect us? Well, there are lots of reasons to care. First of all, an amazing fact, viruses outnumber cellular life by at least 10 to 1 on Earth, at least, probably more. They have the greatest biodiversity of anything on the planet, and they have the greatest genetic diversity of anything on the planet. As we'll see, they undergo enormous genetic variation as they reproduce, and that genetic variability actually benefits other organisms. They can transfer it to their host cells. So they're huge agents of what we call horizontal gene transfer. They drive global cycles. Those 10 to the 30th phages in the oceans, every time they lyse a host, whether it's a bacterium or a protist, they release particulate matter, particulate organic matter that sinks to the bottom of the ocean or it sinks down into areas where it's metabolized and turned over to release carbon and other elements. So they are huge drivers of global cycles. And in fact, without them, I think the Earth would be quite different. We think they're beneficial, so it's good to know what's out there so we can get an idea of which ones are beneficial. And of course, any new pathogen that arises, so if you hear of a new virus arising, in the 1970s it was Ebola virus, we'd never seen it before. It had always been there, it was in animals. They're all out there and waiting to be discovered to jump into people. So that's a good argument to knowing all the diversity that is out there among viruses. Now, if you are overwhelmed and thinking, how am I going to remember all of these different viruses? Well, I would never ask you to do that. But there is one last thought I want to leave you with, which is really helpful in terms of going through the next few sessions. All viral genomes are obligate molecular parasites. They can only function after they replicate in a cell. So a viral genome gets into a cell, it's either RNA or DNA, and it initiates gene expression. We're going to talk all about that because it's really interesting. And the first thing that they have to do is make mRNA. Otherwise, they can't make any proteins. That mRNA has to be translated by host ribosomes. No virus encodes ribosomes. No virus encodes a complete translational machinery. All of them, every one of them that we've discovered, depends on the host cell for translation. The mRNA that the virus makes has to be recognized by host ribosomes. So there's a very intimate relationship between viruses reproducing and the host cell. So next time we are going to use that that simple fact that viruses are parasites of the host translation machinery to order viruses and make sense of all this diversity.